So, what will cancer care look like in 10 years compared to today?
We know that there are different points in the cancer care pathway that AI may be able to have an impact.
You did say synthetic patients.
Synthetic patients, yeah.
Could you just expand on
All of this has to be thought about really carefully in terms of regulation, in terms of ethics, because this is sensitive health data that we're talking about.
At a time in the NHS when only 3% of pathology labs are fully staffed, we really need this kind of automation.
Welcome to the Bryden podcast. Today I'm joined by Dr Lucie Burgess and Dr Emma Colliver from Pharos AI, who are doing some fascinating work with AI in cancer research. And I'll ask Lucie and Emma to introduce themselves and tell us a little bit more about Pharos AI.
Thank you so much for the introduction and invitation to be here today, Adrian. So I'm Lucie Burgess. I'm the Chief Operating Officer of Pharos AI. I have a background in strategy, commercialization, and innovation for data-driven and AI-enabled research. And my role covers our strategy, finance, investment, commercialization, data governance, and our program management team.
That's quite yeah.
It's a broad portfolio.
Yeah, absolutely. And tell us a little bit about the company.
So Pharos AI is a contractual joint venture, so we're a partnership between 4 organisations. We're on a mission to accelerate the path to AI-enabled cancer care. And the partnership brings together 2 world-leading universities, so King's College London and Queen Mary University of London, and 2 NHS trusts, Guy's and St Thomas's NHS Foundation Trust and Barts Health Trust. And with this collaboration, we are bringing together tissue samples from patients who have donated those samples to the trust, and we are digitising those, creating multimodal AI-ready data sets for cancer care to develop new therapeutics, diagnostics and clinical applications.
Thank you. And Emma, would you like to introduce yourself and tell us a little about your role at Pharos AI?
Yes, thank you. I'm Emma, and I'm one of the research fellows at Pharos AI, and I'm working to curate these multimodal data sets that Lucie has just mentioned. So I have a background in physics and then transitioned into working in lung cancer. So now I'm sort of seeing this as bringing these 2 worlds together, that data-driven approach with the cancer research setting, and hoping to bring these data sets to the research community at Pharos.
Well, fantastic. And thanks again for taking the time out of your mission today to talk to us at Bryden Wood. We'd like to learn a bit more about your work. So tell us about the goal of Pharos AI in bringing together large amounts of cancer-related health data, making it easier for researchers and companies to use AI to find better treatments and diagnoses, of course.
So maybe I'll start and then Emma can add. So as I said, we're on a mission to accelerate the path to AI-enabled cancer care, and we're doing that because developing cancer drugs and diagnostics is extraordinarily risky and expensive. So it costs on average somewhere between $1.5 billion and $5 billion to bring a cancer drug to market. It takes somewhere between 15 to 20 years, and there is only a 4% success rate. So those drugs have got to go all the way through from early stage preclinical target discovery, finding the pathways and molecules that might lead to a drug, all the way through to clinical testing. And it's a similar story on the diagnostic side. And in the meantime, people are dying of cancer. So AI holds great promise, we think, in being able to accelerate that pathway and get drugs and diagnostics to market more quickly. So the way that we're doing that is by taking tissue samples of patients that have been collected with their consent through the diagnostic pathway and stored into research active biobanks. And then we're creating multimodal data sets that are AI ready. So spending a lot of time curating the data, cleaning it. We're creating whole genome sequences, RNA sequences, something called spatial transcriptomics, whole slide images of these tumour tissues and healthy tissues and lymph nodes nodes and a variety of other tissues. And then we're linking those to the electronic health records of the patients, which give us a really full, rich, longitudinal picture of the way that they have been treated over time. And then we're putting these data sets on a platform for pharma, biotech, and digital pathology companies to use, and academic researchers to use in their research. And they're using those data sets then, we hope, to create new drugs, therapies, and diagnostics.
And I think you asked, about why AI might be a tool to help us in this. These are, as Lucie said, rich, complex data sets, but, we know that AI can be used to find recurrent patterns, for example, order out of chaos, if you like. And so these data sets are, very complicated to look at for a, human alone or time efficiency wise. We need that tooling to help us on that mission to find those patterns. AI has obviously already had proven impact in other spheres, sort of finding anomalies in fraud detection, sort of lots of image recognition tasks. Can we translate and use that tooling for some of the most complicated data sets out there? And we think, that this is the platform to try and do that on. We'd also say AI in medicine in general is already, showing marked impacts, if you like. We saw in 2024 the Nobel Prize for Chemistry awarded for AlphaFold 2 for that protein structure prediction problem. And that really is underpinning some vast changes now, or transformative changes in the drug discovery and design field. So we'd like to sort of see how else can we harness AI to really improve those patient outcomes ultimately.
And what first convinced you that AI would fundamentally change cancer care rather than just making incremental improvements?
So I think, similar to, for example, we've already talked about AlphaFold. This was a problem that was sort of unsolved for many years. And now this has, really changed the times or the speed over which these drugs can be designed, or perhaps targets can be modeled at the atomic level. So that's sort of one use case. But I think as well, we've seen, from our own group's lab work, from the lab of our CEO, Professor Anita Grigoriadis, and there's a lot of work there on using these digital pathology slides and using AI tooling to try and identify features in these slides to automatically segment or to sort of, outline, if you like, features in these images. And the tooling is proven to have high accuracy when tested against ground truth. And the fact that this could then speed up or create, real efficiencies in clinical workflows is something that I think is excited a lot of people, where there is there are shortages in workforces, for example, in the clinical care pathways. Can AI support in these quite manual endeavours?
I mean, just to add to what Emma has said, we know that at the moment, the way that cancers are diagnosed typically is for a pathologist to look down a microscope at one of these whole slide images, so a section of somebody's tissue. And because of the pattern recognition abilities of AI models, there's an opportunity for AI to be able to do that more accurately and quickly. And it's not just the whole slide images. If we can look at the transcriptome of the individual, of their genomic sequence, that's also something that can help us to understand whether they can respond to a particular drug or a treatment, whether their cancer will spread and metastatize, and potentially their survival rate. So we think this is really exciting. And at a time in the NHS when only 3% of pathology labs are fully staffed, we really need this kind of automation.
And at what point did you realise there was a new venture to be built around this idea?
Well, this was very much down to our CEO, Professor Anita Grigoriadis, and our CTO, Greg Verghese. So they've worked together in digital pathology for a number of years. And one of the things they really struggled with was accessing data from our biobanks, getting that through the approval processes, being able to use it at scale, and then doing all of the work around curation of those digital data sets. So, sending them out for sequencing, scanning the slides, delivering spatial transcriptomics, which is a fairly new tool. And they saw that there was an opportunity to be able to do that more effectively. I mean, what happens right now is that biotech companies will come to our biobanks and they will get access to these tissue slides many times alongside academic researchers. And then they will take those tissue slides away and they'll work on a specific research project which has gone through a really robust data governance process. But then those data sets stay with with the companies that are using them. And what they really wanted to do was firstly to accelerate that process, and then also to democratize it so that when those data sets had been created, they could be made available to everybody for research. So that's the real driver behind this. So Greg and Anita put in an expression of interest in 2023 originally, to a government-backed scheme called the Research Venture Catalyst, which had a very broad brief to unlock innovation in new and exciting ways. And through the application process, they beat off 350 other competitors, and we rewarded funding then as a team in February 2025, and our project started in April.
That's a very interesting background. And what advantages do you get from having formed the venture over research, in a purely academic or clinical setting?
So I think the main driver behind this is to get to sustainability.
Yeah.
So most research projects will be, funded as a research project with a defined start and end, and at the end perhaps they will have created a data set or done some brilliant research or some papers, but they haven't necessarily created a sustainable infrastructure. And the scheme, which was funded by the Department for Science, Innovation and Technology, so thank you very much to our government funders. I have to thank them. Thanks to that, we had a defined goal around sustainability. So one of the things we're really trying to do with Pharos AI is make biobanking more sustainable. So these research active biobanks at the moment are typically either publicly or charitably funded. There are hundreds of them in the UK, thousands across the world, and they do a really important role in collecting tissue samples for use in research, but they don't have a sustainable model. So what we're hoping to do with this, at the moment we are not for profit, but we are exploring models through which we can generate income and revenue and drive that back to our biobanks so that it will sustain their activities over time, because we really think that's important in developing these drugs and diagnostics for patient impact.
So Pharos AI lowers the AI barrier of entry for UK biotechs, drug discovery startups and other high growth companies. What does that mean in practical terms?
Well, I think, myself, I came originally from an academic background. So, for example, a lot of these individual labs may not have themselves the experimentalists on the ground or the funding to do that upfront part of the process of doing the application for and generating the data from the tissue samples, for example. So in that way, those sorts of groups, may be able to access the highly curated AI-ready data sets, which we're hoping to produce at Pharos. So in that sense, this is really allowing them to sort of get a foot in on this answering the questions that they want to, and to pursuing the research that they want to as well. So I'd say that, and then again in terms of this means that it's not at just the provision of the sort of those with the biggest pockets at the start, perhaps, for instance.
Yeah. Do you think this could help create the next generation of UK biotech success stories?
I guess we hope so, don't we, Emma? Absolutely. I mean, just to add to what Emma said, developing these AI-ready data sets is difficult. It involves, a huge amount of work and effort. It's very expensive. So, to create these sequences, to buy the equipment even is very expensive. It's without the reach of most, small AI pathology companies or biotech companies. So by creating these data sets once and making them AI ready, so making them linked, embedded, annotated, all of the things that you need to do to make them available and useful to an AI model. We're hoping to do that once. So we're going very, very deep on certain data sets that we're curating, I should say. We're focusing on breast cancer, lung cancer and pancreatic to start with, because they are all big killers in the UK. And we hope that making those data sets available and, bearing the risk and the cost of that will really then level the playing field for small companies to come in and create new drugs and therapies and some really interesting new models around, for example, survival.
Mm-hmm.
And I think as well, in terms of what you've asked about, do we think this could help to spring up some biotech success stories? We know that AI models are data hungry, if you like.
Hmm.
By working across these two biobanks, we're looking at building this model where we can build those bigger data sets, but in a harmonized way. So rather than each, again, sort of biobank or each research project generating these data sets, but with slightly different language behind it, if you like, we're making sure that from the off, where we can, we're harmonizing this process, which again means that hopefully that number of samples or number of data points that can be fed into or used to train the AI models will be enhanced too.
So do you think that harmonization that could be something would set a template for wider use?
Yeah, I think absolutely, that may well be the ambition of Pharos going forward. We're starting with these two biobanks and these two NHS trusts and sort of working through that process at the moment. But we want to be future-proofing that, thinking how can we make sure this could be rolled out, this model, or could serve as a template for others who might want to follow suit. And that can come at a number of levels. That can come both in terms of any sort of harmonisation that can be done in terms of the tissue sampling and the experimentation on that side. But then at the level of like, what is the shared common language again that we are putting these data sets into the platform in? And so that would be more the sort of role that I, as a bioinformatician, am sort of working on that side of things.
So the interoperability is really important because, again, one of the reasons that it's typically taken so long to bring these data sets together is they're not in the same formats. They kind of don't talk to each other, if you like. So we're doing lots of work around this curation and common data models so that we can bring disparate data sets together from different sites and create these supersets for these AI models that need very large amounts of data.
Yeah, we see exactly the same thing in engineering.
Yeah.
When we trying to move away from humans to machines to look at things, they need that consistency, don't they?
Yeah, and we're using standards. We hope that the community is using things like OMOP, for example.
Thinking about time frames, are we an inflection point, do you think, where AI can speed up cancer drug discovery? How long before you see some real impacts from this work?
Well, I think the jury's a little bit still out on this one, isn't it? There are AI designed or AI optimized drugs now in clinical trials, both for cancer and for other disease types. We have stage 2 candidates, for example, in Silico's IPF candidate that's in stage 2. We do have other companies that have cancer drug candidates now in human trials, if you like. But we don't know the result of those. We know things sort of fail very in late stage trials too. And so as yet, we haven't had that proven model to say that AI has changed that failure rate in a marked way. However, what we have seen is that some of those early stages of the drug design or discovery process, there has been a great speed up, perhaps in the way that those drugs can be designed and put through to those sort of earlier trial phases, pending, of course, passing the safety measures, etc. And so I think that's where we've already seen some movement and some transition. And yeah, we'll look to see the clinical readouts from these trials to really know on that count the difference that can be made. But we also know that AI is having impact not just in the sort of drug discovery or design space, but for example in the diagnostic space and in working, for example, with the readouts or the metrics it can sort of generate from imaging data. For example, medical imaging data is the place where we've already seen 80% of AI sort of medical devices, if you like, in FDA CE approved are in the radiology space. So we know that there are different points in the cancer care pathway that AI may be able to have an impact, and there's already sort of been approval, if you like, for these products.
And what's really interesting then, how some of the advances in the radiology space are translating to pathology, which is very much where we're rooted. So we're seeing a whole breed of new AI digital pathology companies that are developing clinical applications. This is a very different end of the healthcare space. So this is clinical applications being used in hospitals to diagnose certain types of cancer and to look at drug treatment response and survival. Some really interesting examples out there. A company called Ibex Analytics, which is developing a number of clinical applications, had some uptake in NHS trusts. Panacea AI Technologies doing really interesting work in diagnostics. A company called AI4, another called Ataraxis. There are a whole new breed of these companies that are using these whole slide images as well as genomic and transcriptomic signatures from patients to develop new clinical applications that will have direct benefit to patients in the clinic.
So if I understand correctly, the application of AI in diagnostics is likely to have an impact earlier.
It's having an
in
impact
drug
right
discovery.
now.
Yeah.
So, I gave some examples then of companies that have developed clinical applications. They have to go through a safety and efficacy process. So that is led by or managed by different regulators in different countries. But so in the UK, it's the MHRA, is the regulator that decides whether or not these applications are safe and fit for purpose, but they're already being adopted within the diagnostic and prognostic pathway in clinical settings in the UK.
And there are other sort of AI-based approaches, if you like, that are being used in the clinical setting now, quite distinct, if you like, from the sort of biology-centric sort of models we've talked about already, but things like ambient voice technologies being used to help sort of summarize like reporting or patient dialogue and conversation more quickly, tools to help structure reports so that, again, these sort of more bureaucratic, perhaps, parts of or administrative parts of clinical workflows are sort of being addressed by AI tooling. But again, as with all of this, and I'm sure we'll come on to it, all of this has to be thought about really carefully in terms of regulation, in terms of ethics, because this is sensitive health data that we're talking about.
Yeah, absolutely. And those changes in the in both the drug discovery and diagnostics areas, those how do you think that's going to affect the economics of drug discovery, for instance?
Well, I think the hope is that it will drive down costs ultimately because it will help us to test things in silico before they're tested in patients. It will be cheaper and less risky, safer for patients to do that. It will drive down the unit cost of those drugs, and there will be then an impact on the rates that health systems pay for those drugs. But I think we're actually quite a long way off from seeing that come to fruition, both in drug discovery and also in clinical diagnostic applications, because you still have to go through the process of testing in humans. I mean, we're starting to see some really interesting developments. This is, I mean, aside from Pharos AI, we're starting to see things like synthetic patients being used as the control arm for clinical trials, which has great benefit because that way you can give everybody the drug. You don't have to give everybody, you can get rid of those who have to be given a placebo or just the standard of care. They can be given the new drug. So that's really interesting. So ultimately, we hope that
You did say synthetic patients. Right?
Synthetic patients,
Could,
yeah.
could
So
you just expand on on
so
that?
this is using data from patients to generate effectively signatures of patients who are not real. They are a synthetic patients.
Yeah.
So when you have a control arm of a trial, you always have, when you do a drug trial, you have the treatment arm and then either the no treatment arm or the standard of care.
Yeah.
And if you've got patients that haven't responded to standard of care, so they're being tested with a drug that's, a second or a third line treatment,
Yeah.
ideally, you want to give them the hope of the potential new drug. So one of the ways in which you can do that is by generating synthetic patients that you don't treat as the control arm of your trial and giving everybody your new drug, which maximizes their ability to benefit.
Oh, that's very interesting.
It is super
Yeah,
interesting.
that's a new one on me. It does, perhaps conjure up some different images. So, what are the biggest misconceptions investors or partners have about AI-driven biotech?
I think that, there are many misconceptions that AI is sort of maybe a magical tool that's ready to go in all circumstance, or once it's deployed, it will work in sort of all patient cohorts or instances. So I think, this is where having a team of people who are really sort of taking that careful approach to curate our data sets and to look at the distribution of that data here at Pharos AI is important. If you train data on an unrepresentative data set, it may not perform as you expect or give accurate results or readout for a true population. So the idea that AI is sort of going to just be ready to go tomorrow is not necessarily the case. And that's why we need these projects where you're really bringing together the different players who sort of understand the landscape from all perspectives to sort of be part of that journey and to make sure that we're putting this across to the users of the platform.
And I think sometimes there is a misunderstanding of the complexity of, the level of testing and evaluation that needs to take place. This is we go through a very laborious scientific process. We have to do that because we want to make sure that our results are robust. There are new types of AI technologies, of course, that we hope will speed up this process and improve the efficacy of it. So there's a lot of interest at the moment. You might have heard of AI agents that can essentially plot workflows and develop hypotheses. It's fairly early stages for those, but again, we're starting to see some benefit and impact from those.
We've talked about pharmaceutical and biotech companies as being partners. What's the role of big tech in what Pharos AI are doing?
So one of the ways we're working with big tech players for our platform is, Emma mentioned that, of course, developing these models requires compute. So we provide the data and we provide spaces for our customers and researchers to be able to bring in their own AI models and to fine tune and further develop those, or to build models on our platform. And that requires compute power. And one of the advantages of the platform that we're building is to have cloud agnostic compute capability and storage capability with a range of vendors. So of course we're working with all of the the big names to develop that: Google, Microsoft, AWS, and so on. We are working with a lot of smaller companies. We have 2 partnerships with larger companies who are equipment suppliers and service suppliers. So we're working with Tempus. We're using their RNA sequencing technology, which is called Tempus XR. So that's a clinical grade in vitro diagnostic, which we're really excited about, that partnership. And we're also working with a company called 10x Genomics on spatial transcriptomics. And I'll hand over to Emma to explain what spatial transcriptomics is.
Yeah, of course. I think in a minute maybe we'll go into the sorts of data we're producing. In terms of the technology piece, for example, the platform that we're building, we want people to be able to come along to, use these AI-ready data sets, but also to deploy either their own tooling, AI tooling, train their own AI tooling, or use, for example, tools that we've already provided to these end users. And that requires, high-performance computing, for example. So both our sort of Barts Health Trust and sort of Queen Mary's site, and then in our sort of King's College London and Guy's and St Thomas's site, each of those will have their sort of own trusted research environments with high performance computing infrastructure behind it, which the end users will be able to sort of take advantage of, if you like, to run their models and train their models. Another sort of tech player, if you like, that's working alongside Pharos AI at the moment is a group called Flower. And here we're looking in Pharos at thinking, we have these data sets that we're producing from these 2 NHS trusts, but what we can't do, again, is bring these just into their own central repository somewhere. We have to really keep these on their individual sites. But that proves a sort of technical problem, if you like, for people training an AI model who really want to have these data sort of together to do the training in one go, let's say. So this group, Flower, are working with us on what we call sort of federated learning, and building a sort of federated architecture around Pharos, which will mean that the data can remain at the individual sites. So within one trusted research environment over at Queen Mary's and one trusted research environment over on the King's College side, let's say. And Flower will help so that really instead of moving the data, you're moving the models, and able to sort of aggregate the results of those model training. So these are techniques that are sort of starting to be used in other fields as well, but we're really looking forward to working with this partner to sort of build something that's more sustainable, that maybe can be built out even further if other biobanks and trusts were to be coming on board, that keeps that data security and patient privacy right at the heart of what we're doing.
I'm really glad that you mentioned federated learning, because it was literally just in my mind. So this is one of the kind of really exciting new innovative technologies that we're working with to unite these data sets. And as Emma explained, the way that this works is it means that we keep the data sets disparate, within the firewalls of the trust, so that has great privacy preserving benefits, but you can run the analytics over the top. What this means in practice is that actually you've got local models, and then you have a global model that uses the parameters of the local models actually, rather than using the data sets. And we're starting to see a number of initiatives now, national and international initiatives, using federated learning to do privacy preserving analytics, including things like the European health data space.
Yeah, that sounds like a a critical concept for dealing with sensitive data.
Absolutely.
I think so.
And is the volume of data an issue at all?
Not yet, because we're in our fairly early stages. I mean, storage costs are quite high. So these whole slide images are very, very large images. If you think of a,
Yeah,
a typical photograph from a mobile phone might be
yeah.
3 to 5 megabytes. A typical whole slide digital pathology image at high resolution is 11 gigabytes. Yes.
Yeah,
So, I mean, at this stage, we are, we're not going for volume. What we're trying
no.
to do is go, very, very deep, very highly curated, answer specific research questions about specific types of cancer. So I mentioned we're focusing on breast cancer, lung cancer, pancreatic cancer. Of course, each of these have many, many subtypes. But, our vision at the moment is to go deep and make sure those are really well curated, AI ready, rather than very, very broad.
Very
But
broad,
over
yeah.
time, we hope that new biobanks will come on board as partners, as well as our customers in pharma, biotech, and pathology.
It's been suggested that the algorithms are ready, but the real bottleneck is getting hold of the data, which I guess is the whole premise here. How do you approach acquiring, cleaning, and structuring the data?
So we're working with 2 research active biobanks, the King's Health Partners Cancer Biobank and the Barts Cancer Institute Biobank. So these take tissue samples from patients routinely as part of the diagnostic workflow. So they then ask for their consent to take those tissue samples and store them. And the patients give consent for their data to be used for future research purposes, including commercial research, which is really, what underpins all of this. We then go through a really robust data governance process to make sure that the end customers who are using these data sets are compliant, that they support Pharos AI purposes to accelerate the path to AI, enable cancer care, and that the research they're doing is in the interests of the patient.
Yep, and I think from my count as a bioinformatician, we're interested, and once we perhaps have that first tranche of data back from an experiment, we're there to do some quality checks, but also to potentially take it further in terms of the processing pipelines we're putting together. So in terms of quality checks, that might mean if we've sent these or experimentally worked with these tissue samples in batches, is there a batch effect that's really not representing true biological difference, but maybe is a technical difference in the experimental protocol that we need to sort of check for before we're releasing this data, let's say, out into the wild on the platform? And we've got sort of many tools, both standard and there's sort of, tools of research interest to do these sort of QC checks and these quality checks. So that's one of the important parts that we want to do to make sure that the data we're putting on the platform, we've done a sort of series of initial quality checks. We're also using standard tooling to take what might be the initial raw data, if you like, from these experiments and put these into formats that researchers may be more used to using or used to working with. For example, that might be when you do genetic sequencing, instead of taking what is effectively the raw genetic code that comes out, you can use various tools to call mutations in a cancer sample from that. That's comparing a cancer to the patient's, if you like, normal tissue in the body. That can be done in a lot of ways, but we intend to use a standard pipeline across our 2 research sites, so the data put on Queen Mary's and the data put on King's is equivalent in that common data language, has gone through the same processes again.
Can you just expand on what you mean by standard tooling?
Standard tooling. So what I mean here is that we want to create standard operating procedures and protocols, if you like, so that we're using the same tools and versioning of tools on our data, for example, across our 2 sites. Again, as we've mentioned, we're keeping that data within the site where it's being generated, but that means we need to make sure the workflows are equivalent on both. Standard tooling might mean, are there tools that in the research community, for example, have been sort of used routinely or adopted like by sort of many different research partners and institutes? Now, of course, we still want to do our own validation of those tools. We're not just going to take them off the shelf. So we need to go through our own rigorous processes, perhaps on sort of other public data sets and the sort, to be sure that we think that the bioinformatics tools that we're choosing are fit for purpose, if you like. So there are sort of options out there or a short list that we might then look to sort of narrow down our choices from.
And we're also developing our own tools. So one of the tools that the team has developed is something called PySlide, which enables you to do pre-processing for whole slide images for digital pathology, and to do that quickly and easily. And we're making those available, freely available to the community where we can.
In the UK, there's been recent debate on how NHS data should be used. How is Pharos AI approaching patient privacy, consent, and fair monetisation of data?
So all of our data sets are consented by patients, and that's because we have the Human Tissue Act in the UK, which means that whenever you take a tissue sample from a patient, they have to give their consent by law. So that really underpins the use of our data sets. We then create these digital data sets from the tissue samples, and when we get consent from patients who donate their tissue samples to our biobanks, we ask them if they're happy for their data sets to be used for research, including future research, and where we haven't actually stated the purposes at that point, and for commercial research. So that's something that underpins, all of our mission. In England specifically, if you can also apply an opt out for the use of your data for research. So if you can do this through your GP, or if you have the NHS app, you can decide whether or not you want your data to be used for research. So even if our patients have donated a tissue sample and they've ticked the box that says they're willing to use their data for research, we also then apply local opt-out. So if they've said to their consultant or their GP or within the NHS app that they're not comfortable for their data to be used for research, then we will remove their data from the data set. So we want to make sure that, of course, that patients are really comfortable with providing their data sets and supporting this research. We're also doing a lot of work with a group that we've set up called Pivot, which stands for Patient Involvement Voice and Outreach Team. And this is made up of patients who, from our two trusts, who are already involved in patient groups. We're also doing a lot of online outreach to make sure that multiple voices are heard. And we're asking them about their attitudes to, the use of AI tools on their data sets, whether they're comfortable for commercialization, whether they understand the benefits of the sustainability model, and so on, because it's really, really important that we have their support. I mean, clearly, in some areas this can be very contentious. The government at the moment is investing a huge amount of money into the UK health data research platform, which is, so our data is not going into that. It's primarily around electronic health records. The difference there is that that platform will be used without consent because we have an opt-out model in the UK. But if it holds tissue samples by law, you have to have consent, and that's the model that we use.
I would add as well, there are some sort of even quite basic things. This patient data is going to be, is anonymized to the end user. This is not something where people's sort of names and sort of address details are therefore on the platform. So whilst we're building these really quite comprehensive and highly curated data sets, which also align with, important sort of relevant clinical metrics, all of this is done under the view of, making sure that this is not personally identifiable to the patient, for the end user in terms of the patients being used, which is, I guess, critical. I think as well, in terms of the security measures that we're putting around, on the platforms, we're making sure again that there are processes of so that egress and ingress of data onto the platforms is sort of highly sort of regulated and monitored, if you like, by our Pharos AI teams.
So what will cancer care look like in 10 years compared to today?
So we hope it will be faster, better, more efficient, that there will be precision treatments that are personalised to you as an individual. So you'll be able to come into a clinic to have either a biopsy or a blood sample taken, and then to have a very rapid diagnosis which takes into account your personal genomic, transcriptomic, metabolomic, proteomic signature. And get a drug which is designed personally for you, and that will, the diagnosis will happen in minutes rather than weeks or months. The drug might take a bit longer, but you'll be able to get it much more quickly than you currently do now. It'll be less toxic. I mean, so that's on the treatment and care side of things, and on the diagnosis side of things. In the lab, for drug discovery, what we hope is that labs will be fully automated. They'll have much better access to tissue samples and to data. cancer. We'll have AI agents who are finding hypotheses very, very quickly, are finding molecular and genomic targets very, very quickly. And that will lead to drugs being developed within, a much shorter time frame rather than the 15 to 20 years that we're seeing now.
I completely agree that AI has the potential to really sort of be transformative in many different parts of clinical care pathway. I think what we'll need to sort of remains to be sort of focused on is that this must remain patient-centric again, and that the patient is, ultimately the beneficiary of all of these changes, but that this is done in a sympathetic way. And at the moment, we know that there's recently been the National Commission into the Regulation of AI in Healthcare has been instituted in the UK. This is to really think again about this being done responsibly and safely over time, and also with that viewpoint that perhaps AI is an assistant or is an enhancer, but not necessarily a substitute for everything. And I think that's particularly important in a setting where we're talking about people's personal health care as well.
Yeah, absolutely. It's a very hopeful picture that you both paint. What does it mean to you both individually, Lucie?
Well, I'm really proud to work with the team at Pharos AI. For me, it has a real personal mission because both of my parents very sadly died of cancer. My father of acute myeloid leukemia in 2019, my mother of pancreatic cancer in 2023. And I think what was difficult for both of them was quite a late diagnosis, so they didn't get very much in terms of symptoms. Particularly for my mother, she just didn't feel very well for a while. She kept going back to the GP, and it wasn't until really it was, far too late that she was diagnosed with pancreatic cancer, which is notoriously difficult to diagnose and often only shows symptoms in very late stages. So we hope that, these types of data sets and AI technologies will be able to help us find new biomarkers much more quickly and diagnose some of these very late stage cases, because we know that early intervention ultimately leads to treatment and then better outcomes.
Thank you.
And for myself too, my parents have both also been affected by cancer. Unfortunately, I grew up without my father as he passed away when I was just 4. Again, he suffered from cancer, and my mother is on a cancer journey right now. So this is really why I've wanted to direct my research efforts into better understanding these complex diseases and hopefully to improving those patient outcomes with the science that we discover.
Well, thank you very much for sharing your personal connection, and yeah, good luck to Pharos AI and the fantastic work that you're all doing. I'd like to thank you for taking the time out of that mission to talk to us today.
Thank you.
Thank you so much.
Very, very enjoyable, fascinating, and we hope to talk to you again.
Thank you so much.
Thank you
We've
very
really
much.
enjoyed the conversation too.